So how do we get a handle on some of these diseases? I've chosen so far to develop agents against diseases where, again, the genetic basis is understood and that it's monogenic. So I'm not trying to tackle this 7,000 disease landscape that is defined solely on the basis of prevalence, low prevalence. That's important, but it's not the only one. Genetic basis understood. I've emphasized that. The protein misfolding mechanism is an area where we have some special expertise. In many of these diseases are mutations that the protein is formed but has an abnormal sequence so that folding is not appropriate, and so it doesn't traffic normally out of the cell, can accumulate and cause disease. So that's a special consideration that we add to when we diseases when we're thinking of tackling them. Target biology, I've mentioned that. The clinical course being known. We need to understand what the clinical course is if we're going to design rational clinical trials. And for many of these diseases, that's not the case right now. How severe the disease is and the age of the population and the size of the population are important. Unmet needs, that's clearly important, but given what we know about this space, there's so many diseases that need our attention. Many of these diseases have patient advocacy groups. It's been a real eye-opener for me to meet some of these people. They're so passionate, they're so driven. You heard from John and Mary earlier today. It's a real source of inspiration for me. I thought I worked with some sense of urgency, but I feel half dead when I interact with some of these patients and these patient advocates. They're truly the strength, I think, of our segment of the industry. Many of the diseases, where are we? So biomarker and surrogate marker can't be emphasized enough, that we need to know what the drug is doing when we give it to subjects, and if we can tie that to animal data, in vivo data that we've generated during the development of the drug, that's even better and more important. And then surrogate markers tying some endpoint, and Emil made a great example of HIV, to a clinical endpoint, and that was CD4 counts initially, and then it was viral load, but those two surrogate markers changed that disease. And a huge win for our industry and a huge win for the patients with HIV infection. It was done because of the availability of surrogate markers. Here's an endpoint which relates to a clinical endpoint, but we don't have to study thousands of patients in many years to get to that clinical endpoint. We can do it in less time. Many of the kids that enter either the National Clinical Center or Children's Hospital, wherever it's called, and as well as Children's Hospital in Boston, get worked up, and about half of those kids leave without a diagnosis. So clinically, we're still not able to come to grips with many of these diseases, and just in terms of simply diagnosing them into some phenotypic disease. Another feature is a reasonable development path. I didn't put precedent development path because most of these diseases don't have precedent development. We need to be able to guess somewhat so what the FDA is going to expect us to do to get these medicines approved, and is that reasonable in terms of a practical nature of just developing drugs. So all these factors we layer on top of the normal factors that one has when you develop a drug. It's got to be have an appropriate half-life. It's got to be able to be made not all that expensively. It's got to have stability. It's got to have safety, efficacy, etc. So it's a way that we have tried to get our arms around many of these diseases. And to reduce that to practice, we put together a database of the diseases, and then we populated it from different data sources. And then we can screen the database for the different attributes that we're looking for. If we're looking for diseases where there are people working in that area, be it in academia or the biotech community, we have access to pipeline information that everyone does. And so we're able to say, okay, here's either potential competition or potential collaboration. If we're looking to start a program de novo that no one else is working on, we can find that out as well. Whether the disease is described monogenically or not, what the prevalence is, how it's inherited, get an idea of how many publications have occurred around this disease over the recent past, so we get an idea of not only who's working on it, but has there been a significant amount of academic work gone on, the biochemistry involved, has it been described, et cetera, newborn screening, age of onset. So a whole host of features that allow us to winnow down this list of potential diseases. So for Big Pharma, for the Lipitor company, what changes do we have to adapt to? On the one hand, we can say that yes, Pfizer has many primary care of medicines, but on the other hand, I showed you examples of several drugs commercialized within Pfizer that are all in the orphan space. And the example I'd like to emphasize again is hemophilia, that people in that area of the company have been working with those medicines and those patients and those patient groups for many years. And so that expertise we can call on, and it's not foreign to everyone in Pfizer when I hold up my hand and say I want to develop a drug in such and such disease. Clearly, we're dependent on collaboration with biotech and pharma. The last few years, the amount of venture capital funding into the biotech companies has dwindled. It's still present, but it's not as robust as it was. And yet we have maintained or enhanced funding from NIH. So in terms of new ideas, new biology coming from academia, that's as strong as ever. I'm a little concerned with the biotech community as we go down the road over the next few years. Will there be as much development there that clearly we in pharma depend on, and other people have spoken about earlier today. The clinical trials are smaller, and that was discussed in terms of showing safety and efficacy. Surrogate markers, again, in accelerated approval come in. Having to recruit patients from many different clinical sites in different countries brings up the notion of international harmonization. These diseases afflict kids often, and for many of our diseases in industry that we've chosen to tackle have been the more prevalent ones and have been the ones that afflict adults. So that's a different wrinkle. Natural history I spoke about, and then engaging patients and patient advocacy groups is really crucial. They know the patients, they understand them, they know the key experts who are working in the area. They can definitely help us in terms of understanding the disease better and what it takes to get a drug developed. So we clearly are codependent on everyone. I've mentioned the collaborations with biotech. We have a screening program in a particularly rare disease that we're doing with NIH and NCI. Giving industry more incentives for R&D has been talked about a great deal, and I won't belabor that anymore. So in conclusion, I think that there's been a confluence of science and regulatory factors which have brought us all here today and made orphan diseases, rare diseases, genetic diseases a very important and different aspect of the biopharmaceutical industry. I think it's interesting to observe the FDA. And then on the one hand, you've got major consumer groups that are intolerant of any safety issue with any marketed drug. And you've got patient groups that we know to be very, very politically active who are saying, our kids have lethal diseases and no treatments. Please loosen your restrictions. So there's an interesting irony, if you will, that the FDA finds themselves in. And I think if we keep in mind the notion that there's unmet need and the diseases are severe, that we should be able to tailor the regulatory environment intelligently on that basis and not just approach every disease in the same fashion.